Hi and welcome to my OCR A-Level Biology revision with me, Christine. So in today's lesson I'm going to look at lipids. Now, taking you back to GCSE knowledge, we need to understand that if we have a molecule which is covalently bonded, we have to understand that it can either have sharing of electrons that are equal or sharing of electrons that are unequal. Now, carbons and hydrogens share their electrons equally. Therefore, they lack a charge region. And what we say is that a molecule which has lots of carbons and hydrogens are non-polar and therefore hydrophobic. Now, if we look at this glycerol molecule and this fatty acid molecule, we can see quite clearly the glycerol molecule has got a hydroxyl group. So there's 3 hydroxyl groups in a glycerol molecule. And in the fatty acid molecule, we have a carboxyl group. Now, because of the carboxyl group and the hydroxyl group having oxygens and hydrogens, there is actually an uneven sharing of electrons here. Now, this results in what's known as a hydrophilic part to the molecule. So glycerol has got a hydrophilic part where it can dissolve in water because it can form hydrogen bonds with the water. Same with the fatty acid chain. If it's got the carboxyl group, it is able to form hydrogen bonds in water. Now, it's important to note fatty acid chains come with differing lengths. So the longer the hydrocarbon chain, normally we say this is more than 12 carbons, the more insoluble that fatty acid is in water due to its hydrophobic nature. So the monomers, when we look at lipids, we need to understand that it is made up of glycerol and fatty acids. So if we look at the fatty acids then, fatty acids can either be saturated fatty acids. Now what that means is for every single carbon, there is a saturation of hydrogens. So it is single bonds and there are hydrogens found in every place where there is a carbon. Now, when you look at my molecule here, you can see that I've drawn it very basically with this jagged line. Now, every part where there is a joining point represents the position of a carbon. So if we look at what's known as an unsaturated fatty acid then, unsaturated fatty acids don't have hydrogen bonds at every single position. Instead, they have what's known as a carbon double bond. They are missing hydrogens, hence the name unsaturated. So you can have a carbon double bond, and if there is one carbon double bond, we call that a mono unsaturated fatty acid. So you can see quite clearly here, I have 2 different mono unsaturated fatty acids. That's because they have a different number of carbons and they also have a different position in which that double bond is found. And the double bond is shown with the double lines. You can have also what's known as a polyunsaturated fatty acid. So a polyunsaturated fatty acid has more than one carbon double bond in the hydrocarbon chain. Now, it's important to note that those double bonds actually cause the chains to bend. And because it causes the chains to bend, they actually can't pack together as closely. And that's why they're actually found as liquid at room temperature. That's what our oils are. So plants have their fatty acid chains, their lipids, as oils rather than fats that we would class the solid form of the lipid. So if we take a glycerol molecule and we take 3 fatty acid chains and we note that we have our hydroxyl groups here, what we can do is we can actually do a condensation reaction. Remember, a condensation reaction is where you are removing water. In this case, because we have 3 fatty acid chains that we are going to add into our glycerol molecule, we are going to produce 3 water molecules. And by doing that, by removing 3 water molecules, we are joining these 4 monomers together to produce a macromolecule, which is called a triglyceride. Now, it's a triglyceride because it is bonded together with ester bonds. So at each place where the water was removed, we have now formed what's known as an ester bond. And that was formed through a condensation reaction. So another thing that we can do is we can actually create a phospholipid. Now, inside the cytoplasm, there are phosphate ions which are dissolved in the cytoplasm. Now, because they're dissolved, that tells you that they must be polar. So we know that a phosphate ion is made up of phosphorus and oxygen, and we know that it is negatively charged because it has extra electrons. So therefore, a phosphate ion being negatively charged means that it can be dissolved in the cytoplasm. Now, when one of those fatty acids in the triglyceride is replaced by a phosphate group, what we end up with is what's known as a phospholipid. So we have a phosphate attached to the glycerol and two fatty acid chains. We say that the hydrophilic phosphate head, hydrophilic because of those extra electrons in the phosphate, therefore making it hydrophilic, form hydrogen bonds with water. Now, the fatty acid chains, those fatty acid tails, are hydrophobic because they are made up of carbon and hydrogen atoms which are evenly sharing their electrons. So they are hydrophobic. They are a non-polar region. Now, this is important to note. So if we take the phospholipids and we were to place them into water, because of the hydrophilic phosphate head and the hydrophobic fatty acid tails, what happens is those phospholipids will actually form a layer on the surface of water. The heads will be in the water and the tails would be sticking out of the water. Now, they're actually called surfactants because of that. So a surfactant is a type of phospholipid which forms a layer on the surface of water. And that's important when we look at the surfactants inside the lungs. Now, the function of the lipids is mainly a long-term energy store. So what we do is we convert and store our energy in the form of lipids because they are a really good source of energy for respiration, because more energy can be stored per molecule. Now, they are also really important because they are necessary for our membrane function. So the membrane formation is formed because of the hydrophilic heads and the hydrophobic tails. Now, the hydrophilic heads will actually form a way of separating two aqueous solutions by the head sitting on the outside and the tails sitting on the inside. So what we get is we form what's known as a phospholipid bilayer. And that allows for this separation of different solutions. So I will go into that in more detail, so do please check out my biological membranes video. But another thing that is a function of lipids is the stability of the membrane. But to understand that, we need to talk about cholesterol. So a triglyceride is a type of lipid. A phospholipid is a type of lipid. A cholesterol is another type of lipid. Now, a cholesterol has a hydroxyl group, this hydrophilic region. Now, there's only one very small part to it, which is hydrophilic. The majority of the cholesterol molecule is hydrophobic, and therefore that allows it to actually fit inside the membrane, and that causes the stability of the membrane. Again, I'll talk about that in more detail in my membrane lesson, but do please check that one out. Now, another function of lipids is hormone production. Cholesterol is used to manufacture not only vitamin D, but also our steroid hormones. It's also actually necessary for the formation of bile. Now, you'll learn more about bile and steroid hormones when you do module 5 as part of your A level, but it is important to note that without cholesterol, you wouldn't be able to manufacture vitamin D, steroid hormones, or bile. So it's important to note that cholesterol is a type of lipid that is necessary for this formation of the hormone production. Now, another function of lipids is thermal insulation. So we need to reduce heat loss. So therefore, what we tend to do is store our lipids just under the surface of our skin. So that's really important. If you are, for example, an animal that's living in Antarctica, you need that thermal insulation. We also have electrical insulation. So when we look at the neurocommunication module in module 5, please do check out that video. I'll discuss myelin sheath and how that helps in ensuring this electrical insulation. It's necessary for animals who are aquatic animals for buoyancy. So animals like whales need to have a lot of lipids so that they are buoyant when they are living in the aquatic environment. It is necessary for protection of our vital organs. So it cushions our vital organs. As we are moving around, as we're walking around, our organs are having to deal with this movement. And so therefore they are cushioned by having layers of lipids surrounding them. And also waterproofing. So birds, when they are in the water, they need to be able to ensure that they don't carry that water when they fly around. So they have a layer of lipid which waterproofs their feathers. Also plants, their leaves have a waxy cuticle, and that waxy cuticle allows for this waterproofing, so water will run off and fall into the soil so they can then absorb that from the soil. So how do we know if there are lipids present then? So what we need to do is what's called an emulsions test. So the emulsions test is where you're going to mix a sample with ethanol to create a solution. You will then mix that with water and give it a shake. Now, if you mix it and give it a shake with the water, because of that hydrophobic interaction, what's going to happen is you will end up with a white emulsion forming. Now that is going to create a layer on the top, and that layer is going to be white in color, and that is going to indicate that those lipids are present. So if we want to identify the lipids, we will need to do the emulsion test to identify that, and that is a white emulsion forming as a layer on the top, therefore indicating the lipids are present. If the lipids are not present, then the liquid will remain clear. So I hope you liked this video. If you did, then please do click on the like button and subscribe to my channel. Also, if you are looking for a revision platform to test your knowledge, then do check out www.aiqchat.com to help you to revise.